Titration is a quantitative technique that uses a solution of known concentration to react completely with an unknown to determine the unknown concentration or quantity. Typically, the titrant, the solution of known concentration, is added from a burette to the analyte, the solution of unknown concentration, until reaction is complete. Notice that the unknown solution is called an analyte because its chemical formula and volume are known. The concentration is not known. Typically, it is placed in the flask. Usually the titrant, with known concentration, is added from a burette to the analyte until the reaction is complete. This is called the equivalence point. In today's experiment, sodium hydroxide will be the titrant, and hydrochloric acid will be the analyte. The sodium hydroxide will be added to the hydrochloric acid until the equivalence point is reached, which means that all the base and acid have reacted to form water and salt. Often the equivalence point cannot be visualized precisely, so the endpoint is the point at which an indicator changes color. For example, phenolphthalein is an indicator that turns pink in the presence of base, so the endpoint will not be detected until there is an excess base dispensed into the flask. The equivalence point has been passed. It is important that the endpoint occurs very close to the equivalence point to obtain good results. When using phenolphthalein, the paler the pink, the closer the endpoint will be to the equivalence point. So let's get started. You will need the following equipment to perform a titration: a burette, a stand and burette clamp, liquid funnel, the titrant, the analyte and indicator, beakers to hold the titrant, analyte, and waste, an Erlenmeyer flask or beaker in which to titrate the analyte, and a wash bottle of deionized water. The analyte may be in solid form or in aqueous form. If it is in solid form, you will need a balance. If it is in aqueous form, you will need a pipette and pipette bulb. You will also need to mix the reaction constantly, so an electric stirrer and magnetic stir bar are convenient to use. If you don't have these, then you will need to swirl the flask constantly throughout the titration. First, measure the analyte. We have already pipetted the hydrochloric acid solution into an Erlenmeyer flask using a 15 milliliter pipette. If you do not know how to pipette, watch the video titled Using a Volumetric Pipette. Don't forget to condition the pipette before use. Label the flask, cover it with a small watch glass or parafilm, and leave it on the side while you prepare the burette. Place the burette clamp on the stand. If the electric stirrer is small, place it next to the stand and rotate the clamp so that it is over the center of the stirrer. If the electric stirrer is big and the clamp cannot be positioned close to the center of the stirrer, place it on top of the stand. Shift it around until it is stable. If you are right-handed, place the electric stirrer to the right side of the stand. If left-handed, place it on the left hand of the stand. This will ensure that you can reach the stopcock on the burette. Take a closer look at the burette clamp. Notice that the rubber knobs are indented. The burette will be securely in position when it is in between the indentations of the rubber knobs. Squeeze both arms at the same time to open the clamp. Place the burette in between both arms and gently release the arms. Inspect it to make sure it is properly in place and reposition it if necessary. Pour sodium hydroxide into a properly labeled beaker. The experiment should state the volume needed. Cover it with a watch glass to prevent contamination. Let's take a close look at the burette. Notice that the graduations decrease up the length of the burette to show volume dispensed. Consider this example. The location of the bottom of the meniscus before starting the titration is at 21.00 milliliters. This is called the initial volume. Once the titration is complete, we read the meniscus again. It is at 31.00 milliliters. This is called the final volume. The difference between the final volume and the initial volume, 10.00 milliliters, is the volume dispensed. The valve on the burette is closed when it is perpendicular to the body of the burette and open when it is parallel to the body. Now let's condition the burette with sodium hydroxide. First, double check that the valve is closed. Place the funnel on top of the burette and pour a few milliliters of sodium hydroxide into it. Remove the funnel. Don't place it directly on the bench top. Turn the burette on its side and rotate it as you change its angle to rinse the inside walls of the burette with the solution. Place it back in the burette clamp. And empty the solution through the tip into the waste beaker. Repeat this 2 or 3 more times to ensure that your titrant is the only species in the burette. Now let's remove air bubbles from the tip. Close the valve and place the funnel back on top. Add sodium hydroxide until the volume is about the width of your hand. Position the beaker containing sodium hydroxide under the burette. Open the valve so the tip is filled with solution. Don't allow it to completely empty out. Look closely just below the valve. If there is an air bubble, tap the burette to see if it comes out. If not, ask your professor for help. Now let's fill the burette. Adjust the height to make sure you do not have to stretch when pouring. Add sodium hydroxide until it is close to the 0 milliliter mark. Remove the funnel and wait a few seconds to allow drops on the wall to roll down. Adjust the volume if necessary. Don't waste time getting the meniscus exactly at 0. It just needs to be within the graduations. Read and record the value to 2 decimal places. Make sure you are at eye level when reading the meniscus. Add 2 or 3 drops of the indicator into the measured analyte. If using the electric stirrer, hold the Erlenmeyer flask on an angle and slide the magnetic stir bar inside. Today we will demonstrate stirring by hand in case you do not have an electric stirrer. Make sure the tip of the burette is slightly below the top of the flask. Open the valve and allow sodium hydroxide to pour into the Erlenmeyer flask. You might want to add slowly with the first titration so you do not pass the end point. Once you have titrated the first sample and have an idea of the volume needed, you can titrate the second sample more quickly. Sometimes it is helpful to do a rough titration. This means titrating an extra sample first, allowing the titrant to flow quickly. When the sample passes the endpoint, estimate the amount of titrant used, then discard the sample since the endpoint was passed. This allows for an estimate of the endpoint to save time on the actual trials. The data for the rough titration is not recorded anywhere. When the pink color starts to take longer to disappear, add the titrant dropwise. A small drop can reach the desired pink color, whereas a big drop can overshoot the endpoint. If that happens, you will have to discard and repeat the trial. Practice delivering small drops. You can use several methods. One is rotating the valve very quickly. This works best when standing with your hand holding the burette slightly above the valve. Another is obtaining a half drop by closing the valve before the drop falls. Rinse the drop into the flask using the wash bottle. The titration is complete once the pale pink color lasts for at least 15 seconds. Since the amount of analyte left near the end of the titration is small, allow enough time for the sodium hydroxide to find and react with the hydrochloric acid. 15 seconds should be enough stirring time. The goal is to have the palest pink possible that lasts for at least 15 seconds. A beaker with water placed next to the Erlenmeyer can help as a comparison to detect the pale pink. Read and record the final volume on the burette to 2 decimal places. Remember to read it at eye level. Typically, you will want to perform several trials of the same sample in order to obtain good accuracy through averaging. Make sure that all your trials have a pale pink endpoint. When you are finished completely with the burette, place the waste beaker below the tip and open the valve to empty it. Once empty, rinse the burette from the top with your wash bottle. Collect the rinse in the waste beaker. Gently place the burette in the burette canister, tip up with the valve open. It will be properly washed by the stockroom personnel. We hope you have found this video on titration helpful. Be safe and have fun in the chemistry lab.